I'm really excited to be here tonight because I was actually involved in the project myself. And we are celebrating the publication of the great, wonderful book, Sea Creatures in Glass, The Blaschka Marine Animals at Harvard. And so I hope if you haven't seen it, that you will certainly want to see it by the time we finished this evening's festivities and come upstairs later on and purchase one. But we have 4 speakers today who will be shedding light on both the history, the artistry, conservation, and relevance of the Harvard's, the MCZ's collection, Museum of Comparative Zoology's collection of these 430 invertebrate models, which are the center of the book. And this is actually, in some ways, a culmination of a 10-year project, which began under the leadership of Jim Hankin, the director of the MCZ, and Linda Ford, the director of collections operations at the MCZ, to bring together the models, to conserve them, and finally to present them to the public and write this publication. And I would like to acknowledge one person who was not able to be here tonight, Florian Huber, who's one of the authors, and he's actually in Austria, and sadly our budgets would not allow us to fly him in for the evening, but we thank him in his absence for all the wonderful work he did. And of course, any publication like this requires a lot of help and a lot of people. And first, I want to thank, and I know I do this on behalf of both myself and Jim Hankin, the many MCZ curators and curatorial assistants who looked after these models for so many years and cared for them. And we also acknowledge the role of Todd D. Peterson, an alum who generated renewed interest in this collection, which led to the start of this project. And then finally, the exhibition and publication, which were made possible by a generous gift in memory of Melvin R. Seaton, class of 1952. I'm going to introduce each of our speakers very, very, very briefly so I can get off the stage as fast as possible. And each one of them will give a short presentation. After that's concluded, we'll have a tiny break while we move chairs around on the stage, and they're all going to come up and join me on the stage, and so we can have a question and answer session discussion about what you've heard and anything you've ever wanted to know about the Blaschka invertebrate models. So, to our speakers. Our first speaker is Jim Hankin. He's Professor of Biology in the Department of Organismic and Evolutionary Biology, Curator in Herpetology, as well as Alexander Agassi, Professor of Zoology and Director of the Museum of Comparative Zoology here at Harvard. We really like long names at Harvard. Every professor I know has like 4 different titles, and Jim is one of them. Our next speaker is Elizabeth R. Brill, who is our Sea Creatures Preservation Specialist, a very unusual job title. She was trained as a marine biologist, and her father was a glass scientist. So with this background, she is really uniquely skilled for the important job of conserving and restoring the Blaschka marine invertebrates. Our third speaker is David O. Brown, photographer and filmmaker who works worldwide as a producer, writes, and cameraman also, and specializing in marine and aquatic subjects. David is responsible for the magnificent images that form the backbone of this particular book. His work has appeared on NBC, CBS, ABC, CNN, the Discovery Channel, the New York Times, and National Geographic Television, as well as numerous exhibits and publications. And last, but certainly not least, is Gonzalo Giribet, Professor of Organismic and Evolutionary Biology in the Department of Organismic and Evolutionary Biology, and Curator of Invertebrate Zoology, as well as Alexander Agassi, Professor of Zoology in the Museum of Comparative Zoology at Harvard. Okay, so please join me in welcoming our speakers. Thank you.
Thank you, Jane. Hello, everyone. I'm going to begin by telling you a short story about these models. I became the director of the MCZ in around 2001. Now, I am a herpetologist that deals with reptiles and amphibians, and I don't do much with invertebrates, at least here in the museum, but as director, I felt it was important that I get familiar with all of our collections, and I went around to the building and asked the different curatorial associates if they could just introduce me to their collections. And I believe it was when I was in the malacology, mollusk department with Adam Baldinger, who was the curatorial associate, still is the curatorial associate. He's not here, unfortunately, this evening. And he was showing me, it was this classic dark room with shelves all going all the way to the ceiling and dusty and everything that an old museum is in your mind. We're not like that anymore, thankfully. But Adam said, oh yeah, and by the way, you know those people who did the glass flowers? And I said, yes. And he said, well, they also did some invertebrate models, and we've got a few. And, he reached way up to the top and pulled down a cardboard shoebox, literally blew the dust off this thing, opened it up, and there were these amazing models, some of which were not in very good shape. They hadn't been damaged intentionally, but just over decades. They had just been sitting there, and maybe as Elizabeth will explain, they'd started to degrade and fall apart. They were not in the best of shape. But he told me that they had some more models, and I said, wow, this is something of interest. And I started asking around to some of the other invertebrate collections, and they too had a few of these models. So I suggested to Linda Ford, our Director of Collections Operations, I asked her if she could do an inventory of these things. And she said yes, because we didn't have a list of how many we had. These were not databased or anything. And she and her staff did this over the next several months. And they compiled a list of them and started to identify them and take little pictures of them to add to the database. And lo and behold, it turns out we had 430 of these things. And, but again, many of them were at least dusty and dirty, some in worse shape. And we decided, well, we really need to get involved doing some conservation work. And that is how we met, or were put in touch with Elizabeth, who had already done some glass flowers, yes? Glass animals, but elsewhere. So we were put in touch with her. And basically, I don't know, for the last 10 years or so, Elizabeth has been coming here from her home in Corning, New York for a couple of weeks at a time, several times during the year. And she had her little studio, or that's more calling it more impressive than it really was, her desk, in one of the rooms where she had her tools. And gradually she has been restoring them and cleaning them, and she'll have more to say about this in a few minutes. So for me, and then one thing led to another. Todd Peterson, a Harvard alumnus, really was quite jazzed about them, and he had us, or convinced us to loan him some specimens to go to Minneapolis, to the aquarium there. And they've gotten increasingly of interest, and we've actually, as Jane mentioned, had an anonymous donor who agreed to pay for a permanent installation of the models upstairs, and now the book. So it's really amazing for me to think back to that day when I was just in the collection, seeing this dusty cardboard box with some glass pieces, to see what's come of it after these many years. It's really a wonderful experience. So I too would like to thank, as Jane did, all of the staff and people who were involved in both restoring the models, but also in producing the book. Gonzalo and Adam from the invertebrates, among the invertebrate staff, David, Elizabeth, Linda, and so forth. Now, what I'd like to do in the remaining few minutes is just give you a brief synopsis, trying to put the models in the context of 19th century studies and appreciation of natural history. Now, this is an illustration taken from a German textbook in 1865. And it showed that even at the time when these models were being produced in the 1870s and 1880s, this is the state of illustration or understanding of natural history for the public. Beginning in the last half of the 19th century, there was tremendous interest in natural history by the lay public. Now, for some animals that we can maintain alive in captivity in zoos and so forth, it's easier for people to get an understanding of what these animals look like in life and their colors and so forth. For invertebrates, especially marine invertebrates, it's particularly difficult, as I think Gonzalo will talk about. These things, when they don't do well in captivity, especially the marine ones, once they die, they lose all their color. They become often little blobs at the bottom of a jar. They're really not very attractive. And people in the 19th century relied on illustrations such as this, at least in Europe, to understand these models. Now, these are not the most accurate depictions of many of these models. They're almost fanciful, but they give you some idea. The situation was even worse in North America. So, for example, here from 1865 is what is, in effect, a field guide to the marine invertebrates, or many marine organisms, produced by Elizabeth C. Agassiz. She has many claims to fame. Elizabeth Agassiz was the wife of Louis Agassiz, the founder of the Museum of Comparative Zoology, but perhaps more important, she was the founder of Radcliffe College. And Alexander Agassiz was Louis's son. And here they produced this book. It was a massive seller in the last half of the 19th century in New England. And in the preface here, as they state very clearly, this volume is published with the hope of supplying a want often expressed for some seaside book of a popular character describing the marine animals common to our shores. In effect, a field guide. Now, let me show, and then it was in that context that the Blaschkas could enter the scene. This was an available niche for them to produce lifelike, colorful models of marine invertebrates. So here we have the Blaschka family on the right. That's Leopold sitting, the father, Rudolph, the son, Rudolph standing, and there's Leopold's wife, Rudolph's mother, sitting. And there is a copy of a catalog from, I believe, 1870 or so. They sold their models by mail order through, in this case, Ward's Natural Science Establishment. Conveniently enough, the Ward of Ward's Science Establishment was a student at Harvard who worked with Louis Agassiz. And this is how they produced their models in Dresden, Germany, and sold them by mail order. Now, just to show you what an improvement their models were, on the left is an illustration from Agassiz and Agassiz's field guide of a sea anemone. On the right is one of the Blaschka's sea anemones, glass marine animals. Here's another examination. In the lower left, these are a kind of cnidarian. You can see these are so-called stalked jellies. They have 8 different calices. You can see in the lower left, that's an illustration from the Agassiz book. And here is one of in the upper right, is 2 cnidarians, each having 8 stalked 2 stalked jellies, each with 8 calices produced by the Blaschkas. Perhaps the greatest contrast is the Portuguese man of war depicted on the left from Elizabeth and Alexander Agassiz's book, giving no hint of the majesty of the model that we see on the right. And by the way, this model in particular is on display upstairs, and if you haven't seen it, you really do need to see it. It's a spectacular model. Now, from the zoological side, the good times came to an end in 1890, when the Blaschkas were made an offer they couldn't refuse by the Ware family in Boston, who said, if you stop making these silly animals and make only glass flowers for Harvard, we'll take care of you financially for the rest of your lives. And that's exactly what they did. So the dark days began. Now, in fact, there was an attempt to escape from the dark days. Many attempts. In fact, Alexander Agassiz, who was then the director of the Museum of Comparative Zoology, worked through this man, George Goodale, who was the first director of the Botanical Museum, and convinced Goodale to solicit from Rudolph Agassiz an agreement that once he finished producing the glass flowers, would he be willing to undertake to prepare for our museum unique models of some invertebrate animals? The whole museum would then be enriched by your unique work. the Ware collection of plant types, and the Agassiz collection of hitherto unstudied invertebrates. Sadly, Rudolph hung up his tweezers and his glass rods and after he finished the glass flowers and never produced another marine invertebrate glass animal. Thank you very much.
Thank you. This is a very, very exciting night for me. For the 24 years that I've been working on these objects, I've wanted to see a book, and here we go. Here it is. Other people have spouses and children. I have cats and little glass animal friends, and I talk to them all. So I particularly talked to this little sea anemone on the slide on your left. I'm going to get that backwards all night long. I had a little conversation with this guy. I was sitting in a tide pool trying to collect data on corals that were trying to survive in this tide pool. And he kept stinging me, and I think he called all his cousins out, and they started stinging me too. And so I ran back and had lunch, and at the lunch table I met Drew Harvell from Cornell University. And she was in the market for someone to preserve the Cornell collection of Blaschka marine invertebrates. And as a marine field assistant and researcher, and as a lamp worker, I was a really good fit for the job. So for about 17 years, I worked with the collection at Cornell, and while I was at Cornell working with their objects, I got a call from the Museum of Science here in Boston. I restored that collection in about 2 years, and much of it, I think, is still on display. And then I got the call from Linda Ford and Todd Peterson and Jim, and here I am at Harvard, 1,100 objects later. This is pretty much the tools I used for all of them. Cotton swabs, dental tools, soft paint brushes, and some adhesive. Probably the most important tool for me was making my really good eyesight even better. It is no longer, but the fancy little headband in the center of the screen with the light on it was heaven sent. I couldn't have done any of this work without magnification and a lot of light. In the top left-hand corner here, you see an object as I saw it when I took it out of the cabinet. Jim mentioned that these were in storage for many, many years. They were actually still being used. I encountered some professors who were actually still showing these objects to students. Along my way, people would come in my nice little office, my studio, and I'll show you that you just saw it. That table was my office, my studio. The objects, yes, were dusty, but not many of them. I actually had a hard time finding a before and after photo like this one, photos like this one, like these, to show you what a broken object looked like and a finished object looks like. Not many of these objects were broken. Most of them had come apart at the seams because the adhesive that had been used in 1870 was hide glue or fish glue, and it had just aged. So over the years, with humidity and temperature changes, the glue failed and parts came off. These things are mostly assembled with adhesive. So, but in other cases, there are components that are made with really extremely fine lamp working tools. I think there's at least one person in the audience who's going to be pretty interested in these slides. I hope I can make the rest of you interested in them too. In the bottom left, you see the anemone that is the finished restored object. It doesn't really look like very much unless you hold anemones very near and dear to your heart, which I do, even though they sting sometimes. But on the left, you see, I'm going to have to walk away here. This object is the one that I'm really looking at. My father says that looks like an inkwell and I shouldn't be showing you this picture, but they're really good pictures of the little cupcake-like thing, which is what I want you to look at. It's the mouth of the anemone, and in order to make that, whichever one of the two men made this had to start with a tube of glass, fuse other layers of glass onto it, blow a bubble, let the bubble cool, heat half the bubble, and then fold the bubble on top of itself in order to make this wonderful little mouthpiece that goes on the top of the anemone to show you how an anemone eats. The Bloshkas, for a lamp worker today, I don't think you'd find many who would sit down and say, oh yeah, I can whip out 15 of these for you right now. The Bloshkas made these all the time. It's essentially how they made the jellies. The bell of the jelly is a blown bubble that's then sort of folded back on itself. So this very, very sophisticated process makes this sort of dull little anemone very exciting to a lamp worker. At the other end of the spectrum, this siphonophore shows you that the Bloshkas were not afraid to use materials other than glass. These hair-like forms that create the tentacles of the siphonophore are wires that are encased with glass, and each of those threads, so if you follow one down, this one's broken off here, so it's sort of easy to see. If you follow this down, the main wire is covered with glass, but then there's another little component that is another piece of wire with a teardrop of glass on the end. So they made all these parts and then glued them together. And underneath in this colony, there are, I don't know, maybe 40 different pieces of glass that they bundled up and put together up in there too. So the number of pieces of glass that are flame worked and make up one object is often a really large number. X-rays of these objects are a lot of fun. They reveal to us that, as in the top left-hand corner there, you see this little nudibranch crawling across. The opaque parts of that image, the bright white parts of the image, show that that glass has lead in it. It doesn't let, the X-ray shows you that it's lead, and where the glass is sort of just a shadow, it's a soda lime glass. So there are two kinds of glasses at work here. And on its rhinophores, the little curlicue part in the very top left-hand part of the screen, the Bloshkas actually were able to take a piece of soda lime glass and fuse with hot glass, fuse lead glass around that. In glass blowing, that's not something that you can do easily because the coefficient of expansion is different between a lead glass and a soda lime glass. But because of the proportion that they were using of one glass to the other, they were able to pull it off. You can also see in this guy's little anal gill, you can see that there are wires in the parts of his what looks like a tail and isn't really his gills. You can also see that there are wires here in each one of these petals on the sea cucumber, and that's how they would make each of those petals independently and then sort of tie them together like a bouquet at the bottom, stick the mouth on the top. That mouth is made like the boring anemone, and then glue it all together with cotton and stuff and stuff it into the tube that forms the body. So the inside of this part of this sea cucumber right here is really not very pretty at all, but the final result is really magnificent, I think. You can also see the feet of the cucumber in the X-ray on the bottom left. There's a wire that holds those little beads of glass together too, and above that, that speckled area, those are little tiny microspheres. And in the book, here's a teaser for you. In the book, I give an explanation of what microspheres are and how many of them there are. I forget the number. I did the math at the time I wrote the caption. But these little microspheres, they add texture and color. And microspheres are a really interesting, another little interesting glass-making process. You start with powdered glass and just sort of sprinkle it through the flame that's running on the table in front of you. And when it comes out the other side of the flame, it's softened and turned into a sphere. So the heat of the flame softens the glass and makes these little wonderful little microspheres. Sea pansy. I had to put a sea pansy up here. The wonderful, I'm going to run over if I don't shut up. So the really interesting part of these is another glass working process called latticino. So each of those little stalks is worked. The petals are made. The petals are fused to the mouth. The petals in the mouth are glued to the stalk. Then those 30 stalks are glued to the body. The body's painted top and bottom. And so if you think that not a lot of work goes into these things, you're mistaken. Color comes mostly from paint when we're talking about the marine creatures. The flowers are a bit different. There's much more glass in the flowers that has color in the glass itself. Most of the marine invertebrates were made from colorless glass, and then they were painted. And one of the, so I worked on the collection here at Harvard for 10 years. And along the way, I would see things that sort of store away. And if you look carefully at the bottom left-hand image there, there's a texture that looks sort of like concentric circles with little green specks on top. If we look more carefully at these images, you can see that in this nudibranch, these concentric circles appear again, and on the top of that anemone, they're fingerprints. They use their fingerprints in the paint and then put the white dots on over. So we know those fingerprints are intentional because the white dots went on after the fingerprints. And they used what they needed to use to make these things look as lifelike as they knew them to be. And they did whatever it was they could do to make what they thought was a lifelike animal. And there are too many people to thank. But thank all of you for coming tonight, and I hope I've prompted some questions for later. Thanks.
Hello. Thank you very much for being here, and thank you for having me. It's a real pleasure. It's a real thrill. This whole Blaschka world is driven by passion. It's driven by passion for the natural world, and you all share it. We all share it. It's just fantastic to see and experience and be part of. A very special thank you to Elizabeth, because I would not be here without her. I hail from upstate New York, and I was working on a film, which you'll see little snippets of here, about the Cornell collection. And in the course of that, I ran into Elizabeth, was introduced to her by Drew, who's the protagonist in the film. And just thank you so much for involving me here, because the more I learn about the Blaschkas, the more I'm imbued by their passion for absolutely everything alive. That's what drives me. That's what's always driven me. I was telling Jim, I'm a herp nut. I love salamanders. I had every kind of salamander that occurred in upstate New York when I was growing up there. And then I went away for 20 years and came back and was real happy to find that they're still there, a lot of them. And a lot of the organisms that the Blaschkas modeled are still there. And the point of the film that I produced about them is that there's a lot of hope, because where there's life, there's hope, and there's still a lot of life out there. And yeah, we have predicated this incredibly disastrous extinction event that we're all part of, but we also have a whole lot to still work for, and it's just really wonderful. I'm going to show you a little snippet because I'm intrigued. Again, this speaks to the passion that got the Blaschkas involved in the marine world, this little snippet of film.
Imagine a shape. Imagine a color. Imagine virtually any form that life might assume, and you'll find it in the ocean. The sea is home to living diversity that transcends imagination. Diversity that has inspired our creativity and intellect since we first became human. In 1853, master glass artisan Leopold Blaschka was sailing from Europe to the United States. The wind died off the Azores, leaving the ship becalmed for 2 weeks. Jellyfish and other creatures moving past the rail entranced Leopold. He drew them, determined to later capture the shining essence of the creatures in glass.
It's such a simple story, but it's so beautiful. The idea that he's becalmed in a different time when people were moving under different propulsion. He was moving under the wind and the wind deserted him. And instead of going below decks and playing poker, which is probably what most of his fellow passengers did, he stood out on deck and just watched and just opened his heart and his mind and took everything he had and went back, and just for the sheer joy of it, it's my understanding initially, made these wonderful things. And that speaks to the importance of story. in making film, in making images, whether they're still, whether they're moving, it really doesn't matter. My background's in field work. I've come into this not as a, not from the art perspective, but more from the field perspective. That's me over there, another time and place with a charismatic type of charismatic megafauna. These are spotted dolphins off the Bahamas. It's a wild pod that loves to come in and interact with people, or at least they did back then. I don't know if they're still doing that. I shot that in Alaska. That's humpback whales. But I started off Stellwagen Bank, taking people out on whale watches. And again, trying to, I didn't have to do a whole lot with whales, fire their passion for the environment. You know, 40-ton whale breaches, boy, everybody pays attention. Not necessarily the case with a nudibranch crawling by. Most people, even divers, even people who are really tuned into the marine environment, a lot of them just swim right on past. And so the incredible obsession with the little things, with the details, with the magnificent details that comprise life is what I find so intriguing about the Blashkas. And also the timing of the Blashkas. If you think about it, had Leopold been traveling transatlantic maybe 10, 15 years later than he was, he would probably have been under power. He would have probably been propelled by fossil fuel. He would have been burning coal. And they would have steamed right on through and by and past these things, and he never would have seen them. So it was such a pivotal time in human history, in planetary history, as we're finding out. And I think there's a lot to be said about that as well. That's part of my obsession with these guys. It's also interesting, the visual progression from an imaging standpoint. You go from the living thing. I shot this up in Les Escamines. This is the Gulf of St. Lawrence. You can see some metridium, which are these drawn here, and then rendered in glass. So you go from the living thing to a 2D to a 3D. My colleague, the person who turned me on to these critters at Cornell, Drew Harvell, shot these. I can never remember which is the glass and which is the real thing. I have to ask her over and over and over again, Drew, which this is, they're just absolutely perfect, and that's just the wonder of it as well. And of course, the fragility of the organisms, or not the organisms, the fragility of the glass bespeaks the fragility of the organisms and the ecosystems they're part of. And so that's a really important story element. That is the point to me. The fact that, yeah, fossil fuel came along, and sure, it's been a revolution. It's been great. We've gotten a lot out of it. But it's also altered the chemistry and the composition of the ocean. And it's changed. It's the game changer. And so this Blashka renaissance, which we're seeing, because these collections are all over the place, is a really exciting spark of hope, I think, for addressing that. And again, comes down to all about the detail. And you can always take what's called, you can stack focus now. That's pretty easy to do. This is actually about 7 exposures going progressively through different points of focus. And then I stack it in software, and you end up with razor sharp end to end, although I left this deliberately a little tiny bit out just so it didn't look stacked. But, or you can come in on detail. And here, some detail from there and even closer still. And the idea of these guys just spending the time to look and to experience these things and then internalize the detail and render it in glass just blows my mind. And I love it. Every time I work anywhere near any Blashka creation, I feel as if it's kind of carrying on in some small way that kind of passion. So again, here you've got tack sharp, stacked image, an actual shell that they put on glass, and I was just intrigued by the eyeball there. So, and I have to say thank you to Harvard for letting me play with these stills in the book, because I could just do that. They gave me the latitude to do that. It didn't have to be a very literal interpretation. It was both, and it was a whole lot of fun to do. So here's a little bit about the detail from the film.
Blashka models were crafted not just for aesthetics, but to accurately represent individual species. Each color and shape, from red tentacle to blue eye spot, represents underlying biology. Every living form has function, many essential to the survival of the species. And each species has a role in maintaining a healthy ocean.
And that's the key there. We need a healthy ocean. And again, I see this as an opportunity to convey that to people. I actually shot that off the Shoals Marine Lab, which is right up the Massachusetts-Maine border. It's really fascinating to juxtapose the living thing with the glass and go back and forth and play with that. This is a living thing that I shot in Friday Harbor earlier this summer. And there's the piece that's the cover of the bit. And then again, thank you for letting me letting me play with the details, because the form and the color is just exquisite. And then I just want to put out a little quick pitch here. These are global collections. There's an exhibit at Corning Museum of Glass now of the Cornell collection, but they've they've made a digital screen that you can see online, and they've identified over 100 places to which these glass pieces were sent. How the heck they did that at the turn of the last century and got them there intact, I'll never know. But they're as far away as New Zealand. They're in India. They're all over the world. And there are clusters of them. And my hope is to go to where these clusters are and examine the ocean there, look at it now, look at it then, look at the changes, look at the problems, look at the solutions, and basically take the same tack with each of the collections. And I'd love to do that with the collections here and elsewhere in Boston, because there's a couple more. And Stellwagen Bank was not a sanctuary when I was doing the whale watches out there and doing the whale research out there. It is now. And that is one of the single greatest policy hopes that's also gaining a lot of traction right now, is designation of marine sanctuaries where these critters can revive. This is the Dublin Museum. I was just fascinated. This is just serendipity, according to Nigel Moynihan, the keeper of this collection. But these are radiolarians. They're a microscopic organism that they just had been recently discovered. And of course, the Blaschkas went right in there and made them, and made them big enough that people could see. But to see that juxtaposed with a skeleton of a whale was really interesting. And so I was playing with that. Here's a close-up of the radiolarian. For obvious reasons, there aren't a whole lot of these too far away from where they were produced in Dresden. And then while I was there, we went tide pooling. And here's this exquisite beadlet anemone. Here it is in a tide pool. And so again, there's plenty, there's a wonderful wealth of organisms out there still to be looked out for, just as we're looking out for the glass. And obviously, all culture is underlain, the underlying element of that, of culture, is nature, and they go together. They're not separate. And the siloed thought that has separated them is kind of breaking down a little bit. And the Blaschkas just bridged that imaginary divide beautifully. I'm going to close with this.
Leopold Blaschka would have had difficulty imagining the changes to come to the ocean over the next century and a half. We've exponentially expanded human population, knowledge, and capabilities. But in the process, we've put the living diversity of the ocean at risk. The Blaschka models provide a time capsule to measure the ocean of today against that of Leopold's time. As complex and delicate as the glass models are, the living things they represent and the ecosystems these organisms are part of are infinitely more so. Now, scientists, artists, policymakers, everyone has a brief window of opportunity to take the knowledge and tools that we've acquired since Blaschka's time and apply our collective imagination to protecting this priceless, fragile legacy.
Thank you very much.
I'm going to be very brief because, well, for several reasons. It's getting late, and also because I have not been really involved at the level that they have with this exhibit. I'm an invertebrate biologist here at Harvard. We had some of these specimens in our collection. In fact, that beautiful blue man of war that you saw was on display in the invertebrate zoology department, together with the original description and a specimen that they used for modeling it, because we always appreciated that. I do work on invertebrates, and therefore I'm really attracted by a lot of the specimens, and you need to understand also what types of specimens they image, right? Or they made with glass. They didn't do shells in general. They didn't do echinoderms or insects or things that had a hard skeleton that people could actually see, and that they look very similar to what you can find in nature. And instead of that, they went for animals that were not as easy to see or that didn't preserve very well. This is actually a picture taken by Vanessa Knudsen when she was at the California Academy of Sciences. I'm fortunate that she's now working here at Harvard in my lab, actually on these nudibranchs. And many of those beautiful creatures, even today with modern preserving techniques, don't look very attractive, right? We fortunately have photographic techniques to make them look good, but imagine at the time, first when people had never seen them underwater because there were no diving instruments or anything that came below 20, 30 feet. It was actually dredged on the surface. They never saw how those things really looked underwater. So they worked with drawings, and they were trying to represent nice specimens because what they often saw, even in natural history collections, are things like that. So they focus mostly on different groups of animals. This is actually the red coral. This is the first invertebrate illustrated in that book. And you can see the beautiful red texture that is what is used normally in jewelry. This is very common in the Mediterranean cultures, and people have been working with red coral for a long time. But most amazing are actually the polyps, right? These little flower-like, the flesh of the animal that very few people actually saw alive. And probably even the Blaschka didn't see it alive in this case, because what you see in the representation of these red coral Blaschka invertebrates is that all these little flowers are kind of contracted. They're more like ones that are already outside of the water. They really don't have that magisterial open wide flower, right? So they probably got a piece of red coral that was already, disturbed by removing it from the depths where these things live. So these are some of the animals that they illustrated, and they concentrated quite a lot on cnidarians. We've seen a lot of beautiful jellies and man-o'-wars and things like that, and there's many beautiful cnidarians, like these tiny colonial anemone looking things, right? And you only see these great things once they're open. You see the colors, you see the tentacles. Once these things are fixed, they become wide and all contracted. So these are some of the animals that they focus on. And likewise, they look at other things. Even large animals like sea cucumbers, once you get them out of the water and you preserve them, they don't look very pretty. I mean, it's not that they look great when they're out of the water, but some of them have fancy oral tentacles that they use for feeding, like they might get the particles from the sand. And the colors also degrade very rapidly once these animals are outside of the water. And obviously, they looked at different types of annelids. This is one of the feather-dusted worms that only will have this pose once they're alive underwater. Once they're disturbed, they withdraw inside of the tube. They'll never be exposed like these once they're out of the water, even if you dissect them or anything like that. And one of their favorite groups was the non-shell mollusks. So they had beautiful illustrations of cephalopods. These are some of the most beautiful eye-capturing invertebrates that they did. We have a beautiful collection upstairs of many different types of squids and octopuses and nautilus and things like that. And their other big star are nudibranchs. These tiny, animals have beautiful colors. But they also have beautiful folding of the mantle. Actually, there's one of the models of these very closely related species to this one that doesn't look at all like the living thing. What they have is the animal with all these lettuce-like flaps that they have on the side of the animal. When you look actually at the Blaschka model, it looks like a flat, with a little rim around it, because they probably never saw them when they were alive in the water. They only got them, perhaps with the description of the colors, with some of these folds already relaxed when the animal was probably dead. But these are some of the beautiful animals that inspired them. They looked at beautiful shapes of some of these nudibranchs, or the beautiful colors that many of them are able to display with that dorsal flowers they have, their respiratory organs, and those rhinophores that they were talking about earlier with that beautiful spiral-like shape. And to appreciate these things at the time of the Blashkas was almost impossible because there was no photography, because there was no underwater. So that was probably the best picture of an animal that anyone could see. And especially they could ship these things around the world and see a lot of this beauty. And they could use them for studying at the universities. I mean, we can see how here the director of the museum was so interested in getting a good representation of a lot of this biodiversity that otherwise they could only see in 2 dimensions in books, and they didn't look as pretty as they do here. So I think this is a wonderful collection. It's really, it's been wonderful to see them being reconstructed through the years and try to match actually some of those specimens to the living organism. So my only involvement in the book was actually going through the list of the names that we had sometimes since the 19th century and trying to figure out which modern species are those, because names change through time. And a few of those actually we could never figure out which valid species they correspond to. But it's been really nice to look at the pictures of the animals, of the models, try to find some of the animals that might correspond to them. And it's been wonderful to be able to see them every day in the MCZ when I walk to class from my office. So, thank you for your attention.
So I'm going to just throw it open to see if people have questions. I can see one immediately, and please, if it's not clear, then I may repeat the question just because we are filming everything. So, but if you have a specific person or you want to just ask the panel generally, then please go ahead.
Were all the glass marine organisms made in Germany, or were they made here?
So were the organisms made in Germany or made here?
They were all made in Germany. Yes, my mic is on. I don't need that one. Surprise. Yes, they were all made in Germany.
And there was Cornell University also has some?
Yeah, there are lots of, they were marketed all over the world.
So they were selling them to every university, anybody that
They were selling to anyone that knew about them that wanted to buy them, yes. And there was a mail order catalog. Jim showed a picture of the catalog, and there's an illustration in the book. In the United States, Ward Scientific was the distributor, and they were sold in Australia, New Zealand, Japan, all over the States, all over Europe. There's a really big collection at Cornell. I'll keep talking about that. The second largest collection is in Dublin, that I know of, and the third largest collection is here, probably. This is the third largest collection. There's 400, so Blashka people talk in like specific numbers. There are 434 in the collection here. I don't round it off. So yeah, they're all over.
Once they came to America, started making the glass flowers, they never went back to making.
That's right.
That's right?
There were a few years of crossover, weren't there? I think there were a few years of crossover. The first couple of years when they were still working out the contract.
Yes, I should say there are a few glass flower people here, so we do have to be a little bit careful. I'm not going to embarrass them by pointing them out, but they're in the audience.
And
Thank you.
I'd like to add that the flowers are staggering. I mean, they're just amazing. And again, I don't see the need to be too concerned about whether it's a flower or an animal as much as look at what they did. It's just incredible. It's all incredible work.
And where were they working on the glass flowers? At Harvard University or just in Cambridge? They had their own place? Or?
They were all made in Germany too. They were all made in Germany as well.
Oh.
Everything, their studio, they really had a studio.
Yes, they really had a studio.
Unlike
studio.
Elizabeth, their studio
Theirs
was
didn't
in
look that nice.
Dresden, Germany. They made all of their models there and shipped them here.
Oh, so they never did any of this
They
work.
never did any work, and the bench upstairs in the glass flowers exhibit is their bench, is that correct?
It is, yes. There were two. One's at the Corning Museum of Glass and one is here. Yeah.
So you can see it upstairs.
And the drawings from which they made the models that draw first, or that do watercolors, those are at Corning.
Okay, I think we have a question over there.
Could you talk, whoever can speak to this, some more about how they actually developed their zoological expertise? I mean, so they were becalmed for 2 weeks, but could it be the source of?
So the question is about how did the Blaschkas develop their own zoological expertise in addition to the story you told about Leopold?
They did model from the works of Haeckel and Gauss. Haeckel in particular was a noted scientist who made those exquisite two-dimensional works covering a whole lot of the marine inverts. And they were in touch. There's correspondence amongst the Blaschka family and Haeckel.
They worked from drawings of the period. They also they kept aquariums in their studio. I don't have an aquarium in my studio, but they kept aquariums in their studio, and they worked from preserved specimens as well. So they took information in wherever they could find it.
And they were frequent visitors to the Naples Oceanographic Station, Italy as well.
I think this mic is off. So to add also that they might have gone through very different periods because some of the specimens are much more accurate than others. At one point, I think they were modeling just from Haeckel's drawings, which are very idealized. They're not really realistic. And then some of the other specimens are very detailed and realistic. So probably that's the phase where they already had the aquariums in the studio and they were working from live animals. But many were very difficult to maintain in aquariums for a long time. So.
Over there.
Could you talk a bit about more of the restoration process? And I'm especially interested in what it might have taken to decide what the paint colors were and how to match the paints, or what that process was.
I didn't do anything with hot glass or with paint. There were so many objects in the collection that in 8 years, or 8.5 years, it took me every minute that I had to reassemble the objects that had come apart at the seams. And Linda and Jim and I sort of made the decision that what we would do is get all of the objects to the most exhibitable state that we could with the time and resources that we had. So I didn't, I worked very hard, first of all, to stabilize the objects and make sure that any environment they were in or any condition they were in, they weren't going to get in any worse condition. And then of the 434, they all needed to be dusted off, but probably 55 or 60% of them only needed to be dusted off. That other percentage, there were an awful lot of jellies and jelly-like animals that involved lots of those wires. Many of where the wires had been glued onto the bells and the other glass components, those needed to be reattached. So that's where my background in marine science came in. I needed to know what animal had this kind of tentacle. And one of the things that was very interesting when they made glass tentacles, and if you've seen the exhibit upstairs, you'll see a handful of little tiny jellies that they all have glass tentacles, and the glass tentacles are different from object to object, but they're very specifically different. From one jelly to the next, the difference in the length of the tentacle may be 1/8 of an inch, but it's different. And so this tentacle doesn't go on that one because it's 1/8 of an inch longer or shorter. So I spent most of my time dusting them off. I cleaned the plaster plates, which were a huge addition to this collection. When they were shipped here, they didn't come on the plaster plates. Most of the objects were purchased in the 1870s and 1880s here, and they came shipped, and they were mounted on paper boards. It's sort of the weight of a manila folder, paper that weight, and most of them were either glued on or wired onto that paper. And somewhere along the line, I think it was around 1905 or 1910-ish, somebody decided that it was a good idea, and they were right, to put the objects on these plaster plates. So a lot of my restoration involved cleaning the plaster plates because they had become even dustier than the objects in some cases. All right, talking too much.
I'm just curious if the Blaschkas understood the importance of kneeling after they got through making the object.
That's a really good question. Did they understand the importance of annealing?
Do you want to repeat the question?
Yeah, did the Blaschkas as lamp workers appreciate the importance of annealing an object? So when you if you form glass in a fire, you create stress when it cools. I mentioned quickly the coefficient of expansion. If it doesn't cool slowly enough, the stress doesn't go away. So in order to have a glass object that's stable, and in this kind of way, from this kind of stress, you put it in an oven and bring the temperature down slowly to prevent cracking from happening. Most of the Blaschka pieces, the components in almost every piece, I'm kind of winging that number, but just about, I think probably any piece I can think of the glass is so thin that annealing isn't really necessary. So more instability probably comes from the composition of the glass itself if the glass wasn't stable to begin with. So the pieces are so tiny that annealing probably wasn't necessary.
There was one over there at the back.
You have 434 pieces of seed and selection. How many pieces do you have of glass flower selection? And if is it the desire of Harvard to have all those 434 pieces of animals exhibited in the hall like the glass flowers are? And thirdly, how much does Blaschka charge that century or whatever to produce the glass piece of the enamel or glass flower, roughly?
An expensive one in 1870 was $12. Most of them were $1 or $2. I'll let somebody else answer the other questions.
So I think there's someone in the room who can probably give us an absolutely accurate number for the glass flowers. Jenny Brown, stand up and be no longer anonymous.
There are about 4,300 individual objects in the glass flowers.
Are they all on exhibit in that room?
They're not all on exhibit. About half of the collection is currently on display.
Do they rotate it?
A portion of the collection, a portion of the exhibit does rotate, but that's a new thing.
Yeah, so for those of you who couldn't hear, there are about 4,300 objects in the glass flowers collection, about 50% of which are on display, and we have some rotating exhibits too, so look out for the what's coming next? Rotten fruit in about maybe a year, maybe slightly less. Anyhow, in terms of, yes, we would love to exhibit the entire invertebrate collection or a bigger selection. One of the huge challenges for us is the size of the collections here at Harvard. So there are actually 21 million now-ish specimens in the Museum of Comparative Zoology alone. And so I would love to answer your question with a resounding yes. If the dean would give us more space for exhibit galleries, the answer is a resounding yes.
Some of the objects actually need a break once in a while, too.
Yeah, and we do rotate them, I should say that. Yeah, we have about 50, or I think it's 50 or 60 on display right now. So you've been, yeah.
How many years had they working on these species before they started working on glass flowers? How many pieces did they produce for this species?
About 25 years they made glass animals. I think he made the first anemone in 1863 and the last one in 1890. That's about 25. And what was the other part of that question?
How many?
Oh, how many? I don't think any of us know how many.
There was one calculation at a recent symposium in Corning that they were producing one a day for that duration. That's what it added up to in terms of the orders that were received just by tracking the transactions through the Ward's catalog and others.
I'd say that's low.
Julia.
Are academic collections throughout the world still finding their sea creatures, or have they all pretty much been found?
The question is whether everyone has found all the Blaschka sea invertebrate models they have.
So there are small collections at Vassar and Tufts and the University of Wisconsin, and it's either Indianapolis or someplace.
The University of Wisconsin, I think it was 3 or 4 years ago, very similar situation. They found a box on top of a cabinet in an old biology classroom.
And that's what happened to the Museum of Science and Cornell as well. Oh, look what we've got. Let's bring them out on display again. And it's wonderful that nobody bothered when they fell out of use, because it wasn't the best way in the classroom anymore to use a glass model, because you could have a photograph or you could get in the ocean. People didn't just throw them away. They tucked them in a corner instead. And then now they're being treated as art objects, and so the boxes are coming out of storage and the dust is coming off.
So I think we have time for maybe one more question before we all go upstairs. Yes.
Where did the glass, who made the glass?
They did not make the glass themselves. They bought it from sources in Germany and Europe. And I'm not going to get any more specific than that, or I'm going to get in trouble.